Good morning, good afternoon, everyone. I'm Angela with the ProFarm Group, and I'm pleased to welcome you to our webinar today on optimizing the use of ISR and SAR fungicides for crop management. Our webinar today is approved for continuing education units with the Certified Crop Advisors and the California DPR. You can see here on this slide, if you're a part of the Certified Crop Advisors group, you can open your CCA app and sign in to this course using the course code below. And then for California DPR PCAs, we have our course number here. Feel free to screenshot it. I always encourage people to screenshot it just to save it for your records. However, at the end of our webinar, I'll be sharing a different QR code to go to a quiz in which we will capture all of your information along with your license number. And after completion of that quiz and achieving at least a 70% pass rate, you will receive a certificate of completion or a certificate of attendance for this webinar. You need to keep that on file and use it when you're renewing your license. It's important to note that our webinars are not automatically logged with California DPR. You need to make sure to keep that certificate and submit it to the DPR when it's time for your license renewal. Okay. If there are any questions about the continuing education units, please don't hesitate to put the question in the chat or direct them to me. Myself and my colleague Nancy will help you with any of your questions, and you will receive the certificate of attendance by the end of next week. I'm going to go ahead and move forward. If for some reason you need me to come back to this slide for the QR code for Certified Crop Advisors, just let me know in the chat, okay? So today I want to remind everyone that ProFarm Group is a publicly traded company, and any information that we share in this presentation today is strictly for educational purposes. Should you be interested in investing in our company, we encourage you to do your own due diligence before making those decisions. We have some prizes today for some lucky winners. We love to engage with our colleagues, our visitors on our webinars. So throughout the webinar today, my speakers will have a few questions to ask. And if you are the first person to answer correctly, you will receive a little gift package from our company with a variety of the things you see above. So it's just a way for us to say thank you for attending our webinar. And most importantly, we encourage you to engage in the chat, ask questions, correspond with one another. While this is a virtual event, we love to still have that interpersonal interaction. So for our speakers today, our first speaker is Dr. Timothy Johnson. He used to work for ProFarm Group and is now retired and serves as a biopesticide consultant. I want to share a little bit about his background because he truly is one of the experts in biologicals in our country. He has a Bachelor of Science and Master of Science from Iowa State University in entomology, and he received his PhD from Purdue University also in entomology. He has over 35 years of experience in the development of biopesticides, including microbial insecticides, miticides, nematicides, microbial and plant extract fungicides, and pheromones for insect monitoring and mating disruption. He spent over 12 years with Marone Bioinnovations, who is now ProFarm Group, and he was responsible for developing efficacy data worldwide for the company, conducting over 5,000 trials or overseeing over 5,000 trials throughout the world. So, Dr. Tim, thank you so much for being on today. We always appreciate the opportunity to learn from you and hear about your expertise on this particular topic.
Thank you, Angela. That's a very kind introduction.
Our next speaker is my colleague, Brian Guess. He is a territory sales manager in the Southeast. So he covers a variety of states, including Arizona, Colorado, New Mexico, and portions of Southern California. Brian brings a wealth of experience in organic and conventional growing to ProFarm Group and has over 15 years of experience in all aspects of growing operations across multiple firms. He Past experience includes being a director of field operations for live organic produce, where he oversaw all areas of field operations, including the food safety program and vendor compliance systems. Prior to that role, he managed regional grower operations in Southern California for homegrown organic farms, leading all aspects of in-house farm management to ensure the highest dollar per acre return was achieved. Brian also has held roles of increasing responsibility with growers such as Beloean Farms in Fresno, California, and manufacturers such as Helena Chemical. At ProFarm, Brian is expanding the presence of our company throughout the Southeast Territory by building awareness and partnering with both organic and conventional growers and distributors to provide insights and assistance on using biologicals and plant health products in an integrated pest management program. Brian has a Bachelor of Science degree with an emphasis in agricultural business from California State University, Fresno. So Brian, thank you for being on and for bringing this topic to our webinar series this year.
Thank you for the introduction, Angela.
You're welcome. Okay, I'm going to tell you a little bit about ProFarm Group. For some of you, you may be familiar with Marone Bioinnovations, which is what our company was formerly known as. However, last July, we were merged with BioSeries Crop Solutions, and our new name has become the ProFarm Group. So ProFarm Group is the North American arm of BioSeries Crop Solutions, which is based in South America, in Argentina specifically. And if you've worked in global agriculture, you may be familiar with another name, which is Rhizobacter. Rhizobacter has a huge presence in South America, and they are essentially our sister partner. So we, in the North American, our ProFarm, in South America, it's Rhizobacter, and we're all part of the BioSeries Crop Solutions family. So that's a little bit about some of the exciting changes that has happened with our organization over the last year and a half. However, our products remain the same. We have a variety of different products, and our portfolio is expanding rapidly given the merge. We have a variety of products in crop protection, crop health, in crop nutrition. And you can see a variety of them here. Today, we're obviously just going to focus on those that pertain to the ISR SAR topic. Now I'm going to hand it over to Tim Johnson, who's going to dig into ISR, SAR, and help us understand some of the FRAC codes, common ingredients, and best practices when considering these fungicides. So I'll go ahead and hand the controls over to you, Tim. And you now have control.
Thank you, Angela. All righty. Oh, that's a slide advance. I guess I do have control. Well, good morning and good afternoon, everyone, depending on where you're listening in from. And thank you for tuning in as this retired fossil comes out of the closet for an afternoon to help out his colleagues a bit. So my topic today is optimizing the use of SAR ISR fungicides for crop management. And I've kind of divided this into 4 different chapters. One is we're going to take a look at some of the definitions. We're going to dig in a little bit into biochemistry that hopefully won't be too frightening. Kind of take a close look at the FRAC codes. It's not a huge area of active ingredients, but it is growing and continues to grow. I'll talk a little bit about some of the differences and similarities of these active ingredients that fall within this fungicide resistance action committee group. Talk a lot about how to optimize the value, the proper use practices, and then we'll conclude by looking at some examples and talk a little bit about some of the watch-outs, if you will. So definitions will vary if you look in the literature, but generally speaking, SAR, or systemic acquired resistance, oftentimes called the salicylic acid pathway. Basically, whether we're talking SAR or ISR, we're really talking about plant evolution. So if you think about the evolution of plants and animals, there's always something out there that might eat you. Plants can't run, so they had to evolve other means of which to try to defend themselves against plant pathogens. And basically, they went into biochemical warfare, if you will. So SAR, oftentimes it's called the salicylic acid pathway. It's a means by which the plant is triggered to produce a number of plant defensive chemicals. ISR is basically the same goal, if you will, but it has a different biochemical pathway. These pathways are typically activated by jasmonate and ethylene, or jasmonic acid and ethylene. So there's a basic difference there in terms of the biochemical pathway, but at the end of the day, they are oftentimes producing the same defensive biochemicals, and sometimes they even share some of the same genes that are triggered. So the biochemists and geneticists, they like to come up with slides like this to make us remember why we should have paid more attention back in college in biochemistry. And so, if we go back in the literature 20 or 30 years ago, plant researchers were trying to focus in on identifying these chemicals that were involved in this mode of action or these modes of action and the biochemical pathways that produced them. Nowadays, they're really focused more in terms of the genes that are being triggered that produce these biochemical pathways. It's a little more efficient, a more elegant way to see what active ingredients have these modes of action. But at the end of the day, you have a group of active ingredients that trigger the plant to produce biofungicides, if you will. And there's a couple of key points here. One is that since you are triggering the plant, not all plants respond the same to a given active ingredient, even closely related species. Tomatoes and potatoes may respond very differently in terms of exposure to one of these active ingredients. The other difference, of course, is that how long they last will vary depending on the plant. Anywhere from 7 to 14 days, for example. The level of systemicity might differ, for example, and the diseases that they're active against may also differ. So there's a lot of similarities, but there are absolutely distinctive differences between the products. So some key points at the beginning is that by definition, products with a FRAC code P, and that's what I'm going to talk about most of this presentation, do not in and of themselves have any direct contact in vitro type activity against fungal or bacterial pathogens. Therefore, that means they require close attention in terms of the timing, the crop conditions, and the environmental conditions for when they should be applied. And that means for the most part, they are preventative type products that should be applied before the disease is established or the very beginning of pest incidents. If significant pathogen levels have already become established, well, then tank mixing with a contact or systemic fungicide or bactericide is strongly recommended. Now, it's interesting that these active ingredients can come from both synthetic and natural sources. Some of the products that come from natural sources can also be used in both conventional and certified organic production, whereas the synthetic materials are limited to conventional only. So again, a lot of similarity there, but we're going to talk a lot about differences in terms of spectrum of activity, longevity, crop safety, et cetera. So now we're going to take a look at some of the FRAC tables, and we're going to take a look at some of the different active ingredients. So this first one, basically these are FRAC code P type active ingredients. We talk about the target site and code, group name, chemical group, et cetera. The far right are some of the product names. And they're basically listed in chronological order for when the Fungicide Resistance Action Committee accepted them as this mode of action being well established. So the first three, P1, P2, and P3, are all salicylate related chemicals. They are somewhat chemically similar. P1, which is S-benzylar S-methyl, or the active ingredient in Actigard and Bion, was the very first compound that was given FRAC code in this group P. So the group P was essentially founded because of this active ingredient. The active ingredients in P2, P3, and P8 are all somewhat chemically related. These three actives are not sold in the US as far as I can ascertain. They were all discovered by Asian-based companies and are used on rice production, so they've never been brought over to the US. P7 is an interesting group. It's relatively new in this group. It used to be in what was called U33. It has always been a little bit of a mystery as to how the phosphonates actually function, but apparently they now are considered to be FRAC code P type mode of action. Alliet, of course, is a very well-known product, has been around for some time. Phosphorus, acid, and salts are certainly widely used. And it's interesting in that this FRAC code, this P7, is pretty active on oomycetes, which generally is a group of organisms that are pretty tough to control for conventional fungicides as well as biologicals that fall within this FRAC code. So they're pretty unique there. It's also worth noting that this is the one subgroup, if you will, within code P where there has been some resistance observed Otherwise, there has not been any pesticide resistance that has been observed in the field to any of the active ingredients that I'll be talking about in FRAC code P. The second table are biologically derived active ingredients that fall within this group. We have 2 plant extracts. One's a polysaccharide elicitor that is derived from brown microalgae, which we would commonly call seaweed. This is the active ingredient in Vaxaplant. The common name is laminarin. We have a anthraquinone elicitor plant extract that comes from a plant called Renutria satulinensis. This is the active ingredient in Regalia and Pacesetter that I'll be talking quite a bit about. And then we have another subgroup, P6, which are microbial elicitors. So these are microbes that function, again, only through triggering host plant defense induction. And this is kind of an important distinction because we'll talk about some other bacilli in a minute that have multiple modes of action. But we have a product called Lifeguard from Certis USA that is based on Bacillus mycoides. So this particular bacillus isolate functions solely through the code P type mode of action, ISR or SAR. There's also an active ingredient from a species of Saccharomyces, which is a fungi that is a microbial elicitor. I have the product named Julietta here, which is a fairly new product in North America. I may not actually be correct that this product claims the ISR or SAR mode of action. It might be a different Saccharomyces derived product. So I'm still kind of trying to track that down a little bit, if you will. There are some other products that do also induce plant defense resistance, but these are products that have multiple modes of action. So the first group is called BM1, and there's basically there's a couple different groups. One are plant extracts from a plant called Swinglia glutinosa, and then there's others that come from either tea tree oil or mixtures of plant oils. So Timurex Gold has been on the market for some time. It's pretty well known. It has multiple modes of action, including cell membrane disruption, as well as inducing plant defense mechanisms. And the difficulty I find when researching these sorts of products, as well as the BM2 that I'll talk about in a minute, which are biologically derived microbes, is that it's really difficult to tell the contribution of these types of products from the different modes of action. So how much of the mode of action of tea tree oil is from inducing plant defense mechanisms versus cell membrane disruption. That's kind of the point here. So the final group I'll talk about here briefly are biologicals with multiple modes of action that are microbially derived from microbes. So there can be a whole host of different modes of action with microbes. It can be competition for wound sites. It can be mycoparasitism, antibiosis, membrane disruption by fungicidal lipopeptides, which would include our product Stargist, for example, lytic enzymes, and then also induced plant defense. So this includes a number of fungi, such as the trichoderma, that have been well known for quite some time, a number of other fungal species, including Saccharomyces, which I mentioned earlier as a possible FRAC code P-type active ingredient as well, so it must depend on the species or how the product is derived, as well as a well-known number of bacterial species such as Bacillus subtilis, amyloliquefaciens, Nakamura, Pumilus, etc. Some of the product names should be pretty familiar: Serenade, Sonata, Double Nickel, Stargist, etc. I'm sure I left some products off of here. This is a group that has grown quite a bit in recent years. So let's talk a little bit about the active ingredient in Regalia and Pacesetter, Renutria satulinensis. This is a plant extract. This weed is native to Western Asia, or East Asia, excuse me. It is commonly found in North America. Originally was introduced as livestock fodder, and like most introductions, things got out of hand. So it's a pretty widely distributed invasive species on riparian habitats. It is the buckwheat family. It is used as a food in Asian cultures. It is closely related to rhubarb and celery. Also hybridizes with Japanese knotweed that was brought over to the US as an ornamental. And when I used to live in Pennsylvania, you could find Japanese knotweed or a hybrid of Japanese knotweed with giant knotweed growing in a lot of our ditches and a lot of our stream and river banks. It's really quite aggressive. The history is really kind of fascinating in terms of its development as a fungicide. A couple of the key compounds that are found in the plant extract include emodin and physion, which are anthraquinones. The antifungal effect on crops was noted in the mid-1980s by BASF that was looking at natural materials. They did a lot of work with it, a lot of research, a lot of trial work looking at its mode of action, looking at its translaminar effect, systemicity, but eventually they did not commercialize it, but they did kind of spin off the technology, if you will, to two companies, one in Germany, as well as one in the US in the late 90s, KHH Biosciences. Both of those companies developed a product called Milsana. The one in the US was able to get a non-food use label through EPA, and then with the help of the IR4 program, got a tolerance exemption for food uses. That's the good news. The bad news is they were not able to develop a commercial production methodology. In 2008, Marrone Bioinnovations basically acquired that technology from KHH and the labels and so forth. They were successful in developing a commercial production process that was cost effective and introduced the product into the US and are now on about their third generation formulation as they've made continual improvements over the years. And then a couple of years ago, introduced a 12% product in kind of in the Midwestern corn and soybean growing areas called Pace Center. What do we know about it? Well, we actually know a lot because it seemed to be quite the area of interest for a number of researchers in the 1990s, the 2000s, and so forth. So you can look in the literature, and there's easily a couple of dozen different research publications looking at the PR proteins that were being produced on a variety of crops. Focus seemed to be on tobacco, which of course is always sort of the lab rat, if you will, in research. Tomatoes were widely researched, wheat, for example, cucurbits. And basically it's pretty clear that exposing plant foliage to Renutria extract would trigger the production of a number of different biochemicals, especially PR proteins and enzymes that were themselves fungitoxic, usually through the degradation of fungal cell walls or the inhibition of conidial germination, if you will. One of the more interesting crops that was also studied was banana black sigatoka disease, which is kind of interesting, showing inhibiting germination of that quite severe disease organism. One of the other compounds that is also triggered to be produced that was shown in wheat powdery mildew was an accumulation of hydrogen peroxide, which also inhibited the germination of fungal spores for that specific disease. And it's interesting, when I went back and looked at some of the literature for today's presentation, these researchers did note that they did see a bit of an in vitro effect on the germination of Bloomeria spores. And that's actually the first and only time I've seen that in the literature. There are additional effects, and it's interesting in that these groups of products that are derived from plant extracts seem to have more different modes of action, if you will, or additional effects than, say, something that is from a synthetic molecule, if you will, which really isn't surprising. We know when you make a plant extract, you're going to get some additional compounds that are going to come along for the ride. With Renutria extract, we see that plants have an additional accumulation of ferulic acid, which is used in cell wall production, helps the plants produce papillae. And a papillae is a form of lignification, or essentially forming a cell wall, if you will, around a point of penetration by a fungal hypha. So it helps create a physical barrier, if you will, to prevent infection into the leaf tissue. And more interesting, especially when it comes to crop management, and I use the term crop management in my title instead of disease management, is that anecdotally when Regalia was being developed, we would sometimes see people report and see ourselves that plants that were treated with Renutria extract were greener, if you will. We saw plant health effect. So in roughly 2013, 2014, Dr. Diana Cochran, who's now a professor of horticulture at Iowa State University, she was a student at Mississippi State, and Marrone Bioinnovations was interested in the effects of Renutria application on tomatoes and in patients under a variety of conditions, including normal good growing conditions, as well as various stress situations such as drought, cold temperatures, saturation, if you will, in the soil. Really wanted to know if the plants would respond the same when they're under stress as when they're under good growing conditions, because we knew from the experiences with Actigard that sometimes this ISR or SAR mode of action wasn't necessarily triggered when the plants were already stressed. And what we found was that that was somewhat true with Renutria extract, but there were some other effects. And what Dr. Cochran was able to demonstrate in her research was that soluble protein in tomatoes was greater following Regalia application, and leaf chlorophyll content of plants treated with Renutria extract were greater than non-treated. Also, the dry root weight was greater in plants treated with a foliar application of Regalia compared to non-treated plants, a very clear positive plant health effect. So let's talk a little bit about some examples in the field of using these sorts of active ingredients. I've got 4 up here. 2 are really focused on disease control, and 2, although it says disease control and yield enhancement, it's really more on the yield enhancement or plant health biostimulant effects. So we've got fire blight, which is a bacterial disease. It's more severe on eastern apples and western apples. I should have included western apples, but the examples will be out east where higher moisture conditions make fire blight an extremely severe disease. Powdery mildew on grapes, which is a worldwide issue wherever grapes are growing. And then I'll look at some yield enhancement on Midwestern corn as well as California rice. So let's start with fire blight. These are small plot research studies. These are inoculated trials. So in the lower left of each slide, you'll see the details in terms of whether or not an adjuvant was used, what the application dates were, what the crop stage was at the time of application, and when the artificial inoculation was done, as well as when the evaluation was conducted. So this is fire blight incidence on apple blossoms in New York State. We can see in the untreated during the evaluation point, we have 90% of the blossoms infected, so it's a pretty high level of infection. Ag streptomycin, which is an antibiotic, that's the standard, if you will, performed quite nicely. And then we've got Quava, which is copper soap, and coppers are oftentimes used for bacterial disease control. Regalia at 2 quarts by itself, which is kind of a standard rate, if you will. And then Regalia plus a copper sulfate product called CS2005. And we could see that all 4 of the treatments did a very nice job. They're all statistically better than the untreated. And the key really here is proper timing, if you will, to start the applications before the disease came in. So the plant is sort of primed, if you will, before that disease arrives and is already producing those defensive compounds when that fire blight bacteria begins to attack the plant. Those conditions are conducive for growth. So this is example number 1. And this is example number 2. Again, same research in New York State, just the following year. A couple differences. In this case, the Regalia plus copper treatment, where we reduced the rate of Regalia to 1 quart, added the pint of the soluble copper product, numerically was better than the Regalia by itself. It's a very common combination to have those 2 together. Again, the Actigard worked very well. This is on shoot blight instead of blossom blight. And there's one other treatment in here, Firewall, which is an antibiotic rotated with fracture. And shoot blight is an interesting result of a blossom infection. That disease gets into the vascular system of the apple tree. It goes down the shoot. It can go all the way down to the leader. And when that happens, it's very severe. Basically, you have to rogue out the tree. So the grower has to replace that tree. It's physiologically younger than the rest of the grove. And so it's really something to be avoided. And it's very useful to see that a product such as Regalia can be useful for that later season disease in the tree, if you will. So switch gears, go to a powdery mildew on grapes, a classical disease situation. This is with UC Davis, Dr. Escalin in 2022. Very large trial. There's a lot of treatments. They're not all included. These are essentially all biologicals. JMS stylet oil is the standard. The letters A to N refer to the upper left where you see the treatments. Essentially, A, B, C, D means it's applied every week. When you skip a letter, it means it's being applied usually every other week. So it's essentially, we're comparing a 7-day application interval to a 14-day application interval. Obviously, growers prefer a 14-day application interval. Spraying pesticides costs time and money. 14 days is better than 7 days. Sometimes we see a difference, sometimes we don't. So in this study, when the crop was evaluated in late July, after a number of applications, we have about a 41% severity on bunches. It's a good level of pressure. It's not extraordinarily high. And we can see that the regalia, whether it be applied at 7 or 14 days, worked quite well, as did a number of the biological products. So on the right-hand side, we have, at least I know Serafel is a bacillus product. I believe Haller is Pseudomonas. Thea, don't hold me to it, but I believe that's a bacterial. So you can see that really when they're applied properly with the right amount of disease pressure, they did pretty well. The regalia did quite nicely, as did the stylet oil applied weekly. Some nice photographs from the trial. This is when the evaluation was done. You can see the level of pressure in the untreated. We have over 41% severity. So that essentially means 42% of the grapes within a bunch are diseased. So that's a bit of a train wreck. And you can see the grapes looking quite nicely in the various treatments here. So what about plant health? It's an important aspect of these products, especially when they're derived from natural sources. We began looking at this active ingredient on Midwestern corn and soybeans far back as about 2012, I believe. And about 2 years ago, we introduced a formulation specifically for this application called PaceSetter. And essentially what we have seen on corn, soybeans, and wheat especially, we see that by itself, it does control disease. We also see a plant health benefit that typically is seen even when it's used in combination with a synthetic fungicide such as Quadris, azoxystrobin, the original Strobi. And of course, Strobies have been used widely for plant health benefits in Midwestern crops. So this slide basically is a summary of 4 small plot trials. Each location has 6 replications, so each bar is essentially the average of 24 data points. We're seeing about a 7 bushel increase with Quadris alone or PaceSetter alone at a fairly low use rate. And then the combination, we're getting about another 6 or 7 bushels. So it's good to see that these 2 products have plant health benefits from different modes of action, and they appear to be complementary. This slide essentially in the lower left shows the same data, talks a little bit about the return on investment. Obviously, that depends on the value of the crop. It varies from year to year, but for that specific year, which was 2019, averaged a 5.5 to 1 return on investment. So that's really a very good return on what is a pretty modest investment. So we're seeing this product now being used in the Midwest on corn, soybeans, and wheat. I'll show some rice data in a moment. We have quite a bit of rice usage actually in Central America and parts of Latin America. One of my last trips with the company before I retired was in Panama and Colombia, where we're seeing the product be used in rice. And we're also doing some research now on cotton, for example, as well as some other southeastern crops. And it's worth noting that when we're making these applications on these broadacre field crops, we're only making a single application. So this is not something that's being done weekly. This is usually a single application, a tassel on corn, R2, R3 on soybeans, single application on the wheat. So it's very user-friendly. And sometimes we see visual effects. This is a drone photograph where essentially we have a cornfield fungicide only on the right, and we can see down the middle where the PaceSetter was added with the fungicide in the application. Week or two after application, it's quite visual, the additional chlorophyll content in the leaves. Of course, chlorophyll should give us more yield. And we do see this plant health benefit even towards the end of the season. This photograph is taken about 2 weeks before harvest. You can see the green plants on the right with the PaceSetter compared to the fungicide only. But it's important to note that in this case, we don't really see a difference in terms of moisture of the grain. When the grain is mature, it dries down. It's very important to the grower. You don't want to end up with a bunch of extra moisture in the grain that has to be either you get a docked, if you will, at the elevator, or you have to dry it before you take it to the elevator. There's really no meaningful difference between the grain on the right and the grain on the left. Some other crops we've been working with is the southeast. This is peanuts. Plant health effect in this case, we have a fungicide going out on a 14-day interval. This is after 4 applications. We had the grower put in a relatively low rate of regalia with each application. This is before PaceSetter was launched. And you can see the plant health effects merely by the way the rows have closed earlier, which has some additional benefits, not only in having more foliage available to create yield, but it also helps with weed control. And then rice out in California. So we've done a number of trials over the years with Jim Cook at CCFS, looking at adding regalia to quadris for improving rhizoctonia control as well as yield enhancement. This is one of the better trials where we averaged at about a 20% yield improvement. That's on the high side of what we would expect, but we do consistently see an improvement in terms of the yield result. I do see in the chat that we had a question regarding the rate for peanuts. That was 1 quart of the regalia 5. I'm sorry, that was 1 quart of the regalia 5% that was added to the quadris with the peanuts. So that would be equal to I guess it's about a half a liter per hectare or something like that. But anyway, yeah, we should figure that out because I know there's a lot of peanuts that are growing down in Central and Latin America. So liters per hectare, if I remember, 1 quart per acre is equal to 2.37 liters per hectare. But I go back here real quick. Let me make sure on that rate. That might be a pound. It doesn't say, but Juan Carlos, I will double check that. But it probably was a quart of regalia, which again would be 2.37 liters per hectare. So some keys to maximizing the benefits from these products. It's important to note on the label, and I always stress reading the labels, which everybody pretty much has to anyway with all the automated labeling in terms of making your recommendations. But these are interesting products. All of these FRAC code P products, they have a rate that has to fall within a certain amount of water. So percent volume to volume or concentration is important with these products. So you'll see a rate and you'll also see some water recommendations. That's to make sure that they stay within a certain range of dilution that typically relates to crop safety. So you'll see, for example, for both the ActiGuard and the regalia labels, you'll talk about recommending applications basically to actively growing plants that are not stressed. Some plants it's really not particularly important, others it is. I noticed when working on this presentation that the ActiGuard label mentions herbicide injury as a stress, not something we've looked at Marone, but it might be applicable. Again, they're best applied in a preventative program, and they generally provide a moderate level of disease control. It can be a high level of disease control if the pressure is not too high, but if you have disease established, you need to tank mix with a chemical fungicide or another material that has activity on that disease, or use them on a rotation. Both regalia and Vaxaplant claim plant health benefits that can result in yield increases. Not really surprising, since they're both plant extracts, you tend to get a lot of other materials that come along for the ride that have biostimulant benefits. So again, while there are similar modes of action between all these FRAC P active ingredients, there are substantial differences in the diseases that they are most effective against because they are triggering the plant to produce the compounds. They are not acting on the disease itself. So that's why you really have to read the label and make sure that if you've been using one product and you want to try a different FRAC P product, make sure you look at the label and that it's on there and before you go forward. A few cautionary notes when I was reading the labels for these various products. Again, we get back to that plant stress thing, as well as yield issues. The ActiGuard labels mentions, for example, on peppers, don't apply within 7 days of transplant. That's probably trying to avoid a stress situation, nor after beginning of bloom. I'm not sure what that's about. Brassicas and leafy vegetables don't apply when the plants are stressed from drought, cold weather, excessive moisture, herbicide injury. And basically this has to do with when plants are under stress, they actually kind of respond as if they had been treated with an SAR stimulating type herbicide. They tend to accumulate hydrogen peroxide. If you then apply a product on them that stimulates the production of more hydrogen peroxide, you end up with basically the plant eats holes in its own leaves, so you get some crop damage, if you will. One of the interesting things I noted with Actigard is that for lettuce, it's being used for bag purposes, no more than a single application. And for brassicas, no more than 4 consecutive applications. Kind of interesting. Peppers and tomatoes, multiple applications may lead to yield reductions. We don't see that with Regalia. And there are some cautionary notes in terms of Actigard being toxic to fish and aquatic invertebrates. So you have to use some caution there where you apply, and avoiding spray overlap. Again, it has to do with not applying too much of the product from a crop safety point of view. Regalia shares some of the issues in terms of stress when it comes to brassicas and leafy greens. Label talks about not applying the product when extended, unseasonably cold or cold and cloudy conditions are expected when that plant is under stress. Daytime applications are preferred over nighttime applications during those situations. And then there is one other cautionary note about avoiding carrier volumes or adjuvants. And by carrier volumes, it's excessive water volumes and adjuvants that create spray runoff, where you can get drip accumulation at the base of the fruit or the harvested commodity. It's a fairly dark product, and if you end up with everything that you spray to accumulate in a little droplet on the bottom of a grape or stone fruit or something like that, you can basically get something that will sort of discolor the commodity, if you will. So it's really kind of a visual thing. So that's kind of it from that angle. Get my cursor back in the right place here.
Tim, thank you so much. We've got some questions in the chat. Do you mind if we do some Q&A real quick?
No, let's do it now, and then I'll ask my question.
Awesome. Awesome. Okay. So the first question is from Amanda Miller, and she says, have you heard of any work using Regalia on Erwinia and calla lily? We use Regalia on other crops, but haven't tried it on the lilies.
You know, I do not recall that. A lot of the early work with Regalia was on ornamentals before the food use label was granted, but I do not recall any work on lilies. It might be worth going to the IR4 website. They keep track of all the trials they've done over the years. And lilies are one of those, you know, they definitely have one group that works just on ornamentals. They may have done some work in the past.
Great tip.
And then for the second part of Amanda's question, we have a line of Regalia CG specifically for the ornamental market. And we have a treasure trove of data that we've tested within the ornamental, targeting powdery mildew, soil-borne pathogens. So if we'll collect your email after this webinar, and I can kind of send you some of that data for Regalia CG so you can implement it into your operation.
Excellent. There's one more question here from Jeff. It says, Tim, what copper sulfate product did you use in your grape trial that had synergy with Regalia? Berry size and improved chlorophyll looked very obvious.
Yeah, it was CS2005, and which comes from a small company called Magnabon in Florida. That's product is very widely used in the northeastern U.S., used up some of the Pacific Northwest. That was actually a treatment that we did not have in there. That was one that they requested.
Okay.
And I see, Angela, a question about the corn rate and timing. So for the pace setter, the standard rate is 6.7 fluid ounces per acre, and the optimum timing is V8 to like R1 to tassel, or along that period. Pretty much the same as the other products that are going out.
Excellent. Thanks. I missed that question. So appreciate
Yeah.
it. Okay. Any other questions for Tim? Otherwise, he has a question for you all. So I hope you're ready to type in the chat.
Sure. So
go ahead, Tim.
All right. I got to make sure I say this correctly. So during my presentation, I mentioned a popular vegetable related to giant knotweed, Nutria satulinensis, that is often served on a platter with carrots on New Year's Eve. What is it? No, we have a correct answer.
Which one is it?
Celery.
Celery.
Goes good with peanut butter too.
Yeah, it does. So Tom,
I saw somebody answered rhubarb.
Yeah.
Rhubarb. Oh my goodness.
I wouldn't think at the holidays, right?
The rhubarb, yeah. Well, rhubarb's an interesting crop because I think there's like 40 acres of commercial rhubarb in the whole country. But it is also related to Renutria satulinensis. And where I grew up in the Midwest in Iowa, everybody had a little patch of rhubarb next to their house to make rhubarb pie in the spring. Yep. But you had to add a lot of strawberries and 10 pounds of sugar to make it taste good.
Yep. Okay, Tom Shively. So if you don't mind messaging me directly, Tom, with your contact information, so we can mail you the little gift box of items, I'd appreciate it. And of course, you can also, I'm going to type in my email. So if you don't feel comfortable putting it in the chat, you can just email me directly. All right, Tim, thank you so much. Are you able to hang around until the end of the webinar? Or do you have
absolutely.
to run?
Yeah.
Okay.
Yeah. I'm going to, I'll go on mute and kill my video, but I'll be here.
Okay, awesome. So if anybody else has any other questions for Tim, think of them now, and he'll be with us at the end of the webinar for more Q&A. And in the meantime, let's go ahead and have Brian Guest. He's going to give a little bit more detail about one of the market leading SAR biofungicides, the art of use, and some additional field data. So Brian, I'm going to go ahead and hand the slide controls over to you.
Excellent.
And you should be set.
Perfect. Well, thank you, Tim. Always a wealth of information, and I always learn something new from your presentation. So thank you again for that. I'm excited to talk about Regalia. Regalia is one of our commercial products that's available to us here in North America. And by far, this is probably the favorite product of mine in our product portfolio to sell. It definitely performs and exceeds growers' expectations out in the field. There is visible differences the day after you applicate this product, and it's a fun product to sell. So first, why should you incorporate Regalia, or why should you use Regalia in your farming operation? And one of the main points is a high return on investment. This product will improve your yield and overall quality of your finished product. It brings a better plant health perspective to your field by improving disease management control in your IPM program. And it's a flexible and easy product to use. It has a 0-day pre-harvest interval and a 4-hour re-entry, allowing you to spray up to the day of harvest, and gives you flexibilities for if you have to have crews coming into your field for in the leafy greens, weeding or scheduling is one of the common practices. So having that flexibility brings another faucet to the table. And then minimal PPE is required for Regalia. Oops. I'm trying to go to the next slide. There we go. So kind of some of the aspects of it, Tim had touched on these about Regalia. So it has a FRAC code of P5. It is a plant extract of Renutria, or giant knotweed is the common name. The application methods on Regalia, we have a broad encompassing label. So we have verbiage for a foliar application, aerial, and chemigation. You will want to check your specific crop groupings. There are some restrictions, but for the general majority of crops, it is a broad encompassing label. The recommended rates are 1 to 4 quarts per acre. Regalia is a liquid product that mixes easily in the tank and stays suspended in solution. And then the application timing, because it is not a contact kill fungicide, we recommend as a preventative spray or at first signs of disease incidence is when you'll want to apply and use Regalia in your operation. And then kind of some more attributes about Regalia. The tank mix compatibility, there really are no restrictions on what you can mix Regalia with your other synthetic fungicides or your nutritional products. Because it is a plant extract, there is nothing living in the product, so we don't need to keep it alive. And we just recommend when you do mix it in with another product, do a jar test before you mix it in your spray tank. When it comes to adjuvants, we do not recommend the use of any adjuvants with it. It spreads fine on its own and it's absorbed by the plant tissue after application. Water pH of the spray solution, we recommend anywhere from 6.0 to 8.0. And this product is rain fast within 1 hour. And now touching on the modes of action of Regalia, there are 3 primary modes of action that we see Regalia brings to the table. The first one being the inhibiting of pathogen growth through the SAR pathways. Tim touched on the application of Regalia turns on these SAR pathways within the plant cells, and then they're creating these antimicrobial compounds, phenolics, phytolexins, and PR proteins that inhibit pathogen growth and allows the plant to fight off the pathogen infection on a cellular level. The second mode of action is strengthening the cell walls through lignification. The lignin inhibits the pathogen from penetrating into the cell of the plant. And then our third mode of action is it promotes plant growth. We see an increase in chlorophyll the day after Regalia is applied to the plant. And this increase in chlorophyll creates a stronger, healthier plant, which will result in higher and better yields of that treated crop. One thing I love when I use Regalia for the first time with a new grower, I like to have them split half their block, treated versus untreated, and then down to that row, you can see the treated section of Regalia because of that chlorophyll increase. I usually like to go out the next day, walk that field, and you can see that pop of green in that treated section because of that chlorophyll. And that visual effect goes a long way seeing the product working in front of your eyes. And then moving on, how can you use Regalia in your conventional or your non-organic operation? We've created the term BioUnite, and this is kind of tank mixing a biological in with a conventional chemistry. And so we really like the key phrase, harnessing the power of biology with the performance of chemistry. And in my experience with Regalia, since I've been with ProFarm, is there's some key group of synthetic fungicides that really perform well, or there's a synergistic effect when you put Regalia in that tank mix. And so some of these tank mix ideas would be the azoxystrobin class of fungicides. FRAC 11 really benefit from adding Regalia into that spray. Also FRAC 3, quinoxaphene, FRAC 13, also FRAC 7 groupings. And then the Mancozeb line of products, FRAC MO3, are also great products to use in conjunction with Regalia. And then your copper hydroxide or your different copper products, Badge 2X, Quava, are two products that I've had success mixing Regalia in. You have the contact activity of the copper and then the SAR activity of Regalia. So if you are using any of these classes of fungicides in your operation, this is where I would start with mixing in a rate of Regalia in with those fungicide sprays, and you'll see the synergistic effect between the two modes of action really performing and controlling the targeted disease. And so why do we recommend mixing Regalia in with these other fungicides, improving the overall control of the conventional chemistry, helping manage resistance there on the farm? You get more out of your other active ingredients, and also improves the return on investment in the long term. Now I'm going to transition over into some data that follows up these claims and tank mixes that we've seen. These trials are done with accredited universities, and I have a mixture of different crop groups. So you might not be farming this specific crop group, but I want you to kind of see the tank mix or the rotation idea that we used within this trial, and you can kind of learn from that, and maybe you're using one of those fungicides, and you can kind of pull from that and implement that into your specific operation. So for this first trial, it's done with Dr. Pudell Ward down University of Arizona Yuma Extension on lettuce, and we are going after powdery mildew. You can see in the treatment timings, we had 3 treatments, A, B, and C. Moving over to the left side of the graph, you can see the y-axis. We have a disease rating. It was on a scale of 1 to 5. Untreated control was coming in about 2.5, so disease was present in the trial. And then moving on to the purple bar, we did Regalia at 2 quarts per acre, plus Quadris at 12 ounces, tank mixed together at all 3 treatment timings. And the combination of Regalia and Quadris significantly reduced the powdery mildew incidence out in the field. Moving on to the next set of data. It's now into cantaloupes, but it's all still down in Yuma, Arizona with Dr. Pudell Ward, and also going after powdery mildew. Looking at the treatment timings, there were 4 treatments, A, B, C, and D. Also, we had a disease rating scale of 0 to 5, 0 meaning there's no disease present, and 5 being the maximum powdery mildew coverage. On the y-axis, it was the mean severity rating. The brown column, first treatment, untreated control. Disease pressure was high in this trial. We go to the second treatment, Regalia at 2 quarts per acre, tank mixed with Quintex at 4 fluid ounces per acre, applied at all 4 treatment timings. We had 100% control of powdery mildew in this trial. And you can see there were 3 other applications in this trial, all done at the 4 treatment timings: Serafel, Serenade, and then Cinerate mixed with Aura Boost. So this just shows the combination of Regalia and Quintex performs very well on powdery mildew. Okay, now moving on to a different crop in a different region. This is on pumpkins with the University of Illinois. We are looking at the Xanthromonas. And so how we measured this trial is we have the green bars is the percent fruit with bacterial spot, and then the blue bars with percent rotted fruit. And you can see as an untreated control, disease pressure is high with the Xanthromonas and bacterial spot manifesting in the fruit. Looking at this first treatment, we did it a little different. It wasn't a tank mix of Regalia. We did a rotational. The Manzate at 1.5 pounds plus Coside at 1.5 pounds alternated with Regalia with 1 quart per acre. And that's tank mixing is one option, and then also a rotational, incorporating Regalia into a rotational IPM spray program is also another option that you can do on your farm. And as you can see, the Regalia rotational treatment offered substantial control. We had Lifeguard standalone treatment, and then we did a Manzate Coside alternate with Lifeguard also in this trial to compare. Moving on to squash with the University of Florida. Also, we targeted powdery mildew. On the y-axis on the far left of the screen, average disease severity is the measurement. The brown column, water treated check, so disease was present in the field. We did Procure at 8 ounces standalone. And then we did a Regalia alternated with Procure rotational spray that offered substantial control, about 70% less powdery mildew in this trial, alternating Regalia in with a Procure treatment. And then moving on to our last crop grouping, I wanted to include almonds. Almonds are a big area where Regalia has a strong fit. And so this one we went for almond scab was our targeted product, or a targeted disease. We had 2 treatments. One treatment was at petal fall, and then the second treatment was 2 weeks after petal fall. And on the y-axis, the far left of the screen, we had lesions per 100 leaves was the measurement, and then we took 3 sampling times, the blue column being the first sampling, green being the second, and black being the final 3rd sample. And looking at the untreated control, you can see the disease is present in the field. We did a bound treatment at 12 fluid ounces, both treatments. And then we did a quadris top at 6 fluid ounces mixed in with Regalia at 1 pint per acre on both treatments. So this would be a tank mix example of how you can use Regalia. We see substantially or even control with the bound, and it had a higher ROI when you looked at the costs of each product side by side. And then moving on to our last data set, this is also in almonds. So this is going after blossom, brown rot blossom blight. And the measurements we did percent incidence in the field. And then scrolling all the way over to the far right, you can see the Fontellus, the very last treatment on the right-hand side. We did Fontellus at 20 fluid ounces per acre on A and C applications, and Regalia at 2 quarts per acre on the B application. So this was not a tank mix of Regalia. This was in a rotational program. And the Fontellus and Regalia rotational program delivered just as good as control as your Quadris Top or your Turmerex Gold application. And that concludes the data slides for me. I just wanted to kind of show you the different ways we have used Regalia successfully in a rotational or in a tank mix program, and how you can incorporate it potentially into your specific operation. If you have specific questions for your crop grouping in your region, please reach out to your local territory sales manager, and they can kind of walk you through specific tailored rates, timing, and nuances. But this was just kind of a general overview of how it's being used successfully, how I've seen it used, and the success stories. And moving on, this is a link to our website. We have other resources on our website that go into depth about our other product portfolios, emerging diseases or pests, and how we can help in controlling those new pathogens. And then we also have a slew of technically based flyers and marketing product, how you can incorporate our products into your specific crop grouping. And that can be found on our website or the link here. And then here is a list of our sales team throughout North America. So I'll give you a minute to write down the phone number and email of your specific sales rep in your region. And then Angela, did you want me to pass it back to you?
Sure. So Brian, there are some questions.
Okay.
The first question is, can you or would you recommend mixing Regalia with a FRAC BM02 fungicide?
Yes, that is. The BMO2s are also triggering the ISR and SAR pathways. We're not really sure which pathways the BMO2s and Regalia are, but we have mixed the bacillus-based fungicides with Regalia and have had good success. And you could also alternate. A rotational between the two might be another way to use it. And then I see, does the copper compound matter significantly, sulfate versus other coppers? You know, I've had good success with Badge 2X and Regalia. I like the copper sulfates and copper hydroxides. I think there's a product out there that has the two of them mixed. I'm not sure if that's Badge 2X or Coside, but I think the different kinds of copper will provide different modes of action on the control, would be my assumption with the copper-based products. But Tim might have more insight on copper.
Yeah, I mean, you mentioned the badge, which, you know, Regalia is, we've done a lot of trial work over the years with it. It mixes nicely, seems to work. I don't think from a technical point of view, there's any reason for us to believe that there is a difference in terms of the copper source. Same thing with the CS2005. We've done a lot of work with those folks in the Northeast and Upper Midwest over the years, and it's worked well. But, you know, from a technical point of view, there's really no reason to believe one works better than the other. Just as so many of them, you can't develop data with all of them.
All right, excellent. Thank you, Tim and Brian. If there are any other questions, please put them in the chat now. We're right at time. Gentlemen, bravo, and like perfect timing to the webinar. Just a couple of more contacts in case you need them. We have our field development staff, a little bit more technical expertise here. So feel free to screenshot this. We'll also be sending out a PDF copy of their presentation to all those who attended. And I will come here to the final slide. This is the slide that you can scan for taking the quiz. So if you are trying to receive credits, especially with the California DPR, you must fill out this quiz, and that will provide us with the information we need so we can provide you with a certificate of attendance. If you don't get a 70% or greater score, just let us know, contact me, and we'll give you a different quiz to try to take so you can get that credit. Let's just double check and make sure. I think I've also had success with Regalia and Coside for walnut blight. Thank you, Jake, for chiming in. And then one more question. Have you ever used Regalia to control omicite fungi?
Oomycetes.
Oomycetes.
So that's like leaf blight, downy mildew. So that's a great question. Oomycetes are more difficult to control. And so we have looked at Regalia on quite a few of them. Definitely worth taking a look at the label. Some cases we've had pretty good success, some cases definitely less so. So one where we have had good success, for example, is downy mildew on greenhouse cucurbits, where it seems to work better in a greenhouse environment than outside. I have no idea why. The one where we have not had particularly good success is like late blight on potatoes, for example. And it seems like potatoes just don't respond as heavily to regalias as we might hope. So definitely look at the label. There should be more specifics in terms of which ones it makes sense. Probably definitely more of a tank mix situation than an early standalone. Oomycetes are tough. We kind of refer to Stargus as more of our oomycete type material. And we have another compound in development that's a few years out that we're pretty optimistic about.
And Sebastian, I would, if it's an organic operation, mixing with a contact kill fungicide, a parasitic acid, we have a Jet Ag product, and Ag and Regalia are an excellent combination. That contact activity of the PAA will control those oomyces, and then the ISR with Regalia will just build that immune system for that plant to fight off any oomyces that are still left in there. That could be an interesting tank mix you could try out too. Or even Jet Ag and Stargus would also be another option too. We recommend.
All right, for those, Brian, do you have a question for the team?
Yes, so my question.
Okay, I forgot about that. I'm so sorry. So for those that are still hanging on at the very tail end, there's a chance to win one more prize. So go ahead, Brian.
So what is the common name for the active ingredient in Regalia? Not the scientific name, the simple common name. It's been referenced a couple times. There are some pictures.
There we go.
It should be that's the full common name. There we go, giant knotweed.
Oh, trick question. All right, so David Cover, is that correct, or Cover?
Looks like
David?
he is the winner.
All right, so David, if you don't mind messaging me privately on the chat, or again, I put my email in the chat earlier. You can just email me directly with your mailing address. Do we have any final questions for our speakers today? We'll hang on for just a few more minutes. If any final questions pop up, please don't hesitate to put them in the chat. Otherwise, you know how to reach everyone. You can also just visit our website and go to up on the upper right-hand corner, talk to a crop consultant or talk to a crop expert. If you click on that, it will provide the team and all of their contact information as well. So our website's a good resource for you. With that, I will just remind you one more time, if you're attending to receive credit, please make sure you take the survey, the quiz, and we will be following up with all those who did complete the quiz by next week, Thursday, with your certificate of attendance. We have had a couple of experiences where emails didn't go through or they weren't received. So please don't hesitate to reach out if you do not receive your certificate of attendance. And with that, thank you so much for attending our webinar today. We appreciate you joining us, and we look forward to hopefully seeing you on a future webinar. Have a great day, everyone.
Thank you.